In the second block of this lecture regarding statistical inference, I want to talk about hypothesis testing and statistical power for the hypothesis tests. Here's the overview. First of all, I want to talk about hypothesis testing, and then the power and sample size for hypothesis tests. Hypothesis testing. The goal for hypothesis testing is to test for a scientific question, and we need to translate the scientific question into a hypothesis. In terms of statistical hypothesis testing, we need to have two types of hypotheses. The first type of hypothesis is called null hypothesis, usually be denoted as H sub 0. The null hypothesis is a general statement with default position that there is no relationship between two measurements or there's no association among different groups. For example, the null hypothesis can be there is no difference in mean of blood pressure between men and women. The other type of hypothesis is called alternative hypothesis. Usually it's denoted by H sub 1 or H sub A. This is the hypothesis to be accepted when the null hypothesis is rejected by a hypothesis test. Here is an example of the hypothesis. In this example, we want to look at the association between non-small cell lung cancer and a microRNA called MRI-21. The scientific question is, in order to investigate the relationship between the microRNA MIR21 and non-small cell lung cancer, we hypothesize that patients with diagnosed non-small cell lung cancer will have elevated expression of the microRNA called MRI-21. The study design is to compare the MIR-21 expression between subjects with diagnosed non-small lung cancer and healthy controls. And then we can have these two types of hypotheses. The null hypothesis, H0, will be the mean of MRI-21 expression for patients diagnosed with non-small cell lung cancer is the same as the mean of MRI-21 expression for healthy controls. So basically they're saying there's no difference in mean of the MRI-21 expression between the healthy control and the patient diagnosed with non-small cell lung cancer. In terms of alternative hypothesis, there are two types. The first type, we can say that the mean MR21 expression for patient diagnosed with non-small cell lung cancer is greater than that for healthy subject. So this is lead to a one-sided statistical test as the alternative hypothesis is greater than. The reason you can have this type of hypothesis is your scientific question or your scientific hypothesis is that patients diagnosed with non-small cell lung cancer have elevated expression in this microRNA. So you can do this hypothesis and use a one-sided test to test the null hypothesis. Another type of alternative hypothesis, H1, could be that the mean of MRI-21 expression for patients diagnosed with non-small cell lung cancer is not equal to that for the healthy subjects. This will lead to the two-sided test as alternative hypothesis is not equal. So not equal can mean less than or greater than. This type of alternative hypothesis is usually used when you do not know the direction. That is to say, you don't know for patient diagnosed with the non-small cell lung cancer, should their MRI-21 expression be elevated or reduced. Here are two types of errors in hypothesis tests, and these errors can be used for setting the statistical test level, or called alpha level, or later on we'll talk about power. The second type, or the type 2 error, beta, is related to the power we'll talk about later. The first type of error is called type 1 error, and usually we use an alpha to denote that. So the type 1 error is the probability of rejecting the null hypothesis given it is actually true. This is the false positive rate. Type 2 error usually be denoted by beta. This is the probability of failing to reject null hypothesis H0 given it is false. In other words, this is the false negative rate. P-values. In hypothesis tests, you usually see p-values. What is a p-value? In statistics, p-value is the type 1 error that a hypothesis test will have when rejecting the null hypothesis H0. Note that a p-value is the probability for the data as if H0 were true, instead of the probability about the hypothesis itself. The characteristic of p-values. First of all, a small p-value indicates that observed data were an unusual event if H0 were true. So that is to say, if you have a p-value, you have enough reason, at least statistical reason, to reject the null hypothesis H0. The second characteristic is that the smaller the p-value, the greater the evidence against H0. The third one is a larger or large p-value only implies data could be consistent with H0, but not necessarily H0 is true. In a lot of situations, such as if your hypothesis test lose power, you still can see a large p-value, but this not necessarily mean that H0 is not true. A hypothesis testing. In order to do the hypothesis test, we need some simple decision rule for rejecting the null hypothesis, and therefore you can say we reject null hypothesis and we can use the alternative hypothesis to help us make conclusions. First of all, the simple rule requires you to pick a nominal error, and this error is usually called test level or alpha level. And this is for which you are comfortable with. So usually, in general, a custom is to use 0.05 as the test level. And this is a highly or very commonly used alpha value. Then you can decide to reject null hypothesis only if the p-value is less than or equal to the alpha value being picked. That is to say, if you have your p-values less than 0.05 in this example, you can claim you reject null hypothesis. Otherwise, you will say you fail to reject null hypothesis, and you can accept the null hypothesis and help you to make conclusion. For this rule, we can guarantee that the type 1 error, which is explained on the next slides, will be generally less than or equal to the test level. Here is one example showing usually how people report the parameters from hypothesis tests. This is a paper from New England Journal where the author want to compare two different types of therapy, the endoscopic sclerotherapy with the endoscopic ligation. Here are the hypotheses in their literature. The null hypothesis is there is no difference in effectiveness or safety outcomes between these two different types of therapy, the sclerotherapy or the ligation. alternative hypothesis is there is a difference in effectiveness and safety outcomes between these two different types of therapy. Here is how they report the statistical methodology and the sample size related to the power. First of all, in the second paragraph, the authors say statistical students' t-test was used to compare the means of continuous variables. The Wilcoxon rank-sum test were used for skewed with ordinal data, and the chi-square test were used for discrete variables. And this is usually the methodology they use for conducting the statistical hypothesis tests. And then in the first paragraph, they say, for two-sided tests to be used, 170 patients were required, and this number were from their power population. That is to say, they need certain sample size in order to get enough power for the tests. And then they mention patient accrual and the major endpoint were reviewed at 12 months intervals. Significant difference in the incidence or complication and a significant increase in mortality in one cohort were found at 24 months review after enrollment of 130 patients, and then enrollment in the study was therefore terminated. And this is to say, with a smaller sample size, they already be able to detect a signal, and therefore they do not have to recruit more patients. And this is somehow related to the sample size and power as well, but this is something special called data monitoring. In this slide, we can see how they report their results. Table 1 are usually used for report patient characteristics. So if we zoom in to the table 1, and you can see they report all these continuous measurements by two different types of therapies. And they report the number of patients in each of the therapies. Because this is a randomized trial, they have relatively balanced design, and they have 65 in the sclerotherapy and 64 in the ligation. And they report the mean plus standard deviation for the age, and mean plus minus standard deviation for different kind of scores. And the table 2, in this table they report the effectiveness parameters, such as the number of active bleeding at index treatment, and they can report the frequency and the percentage. And they also report mean plus minus standard deviation. And then based on this, they have the p-values reported at the end of the table. For example, they say p-value equal to 0.056 for comparing between groups for a specific parameters they report. And this is to say they can conduct a two-sample t-test to comparing the mean, and they report the p-value corresponding to that hypothesis testing. So remember, in the hypothesis testing, the null hypothesis is there's no difference between the two groups. And the alternative hypothesis is there is a difference between the two groups. Lastly, I want to talk about the power and sample sizes. The statistical power of a hypothesis test is the probability that the decision rule of the test will reject null hypothesis when the null hypothesis is false. Note that the power will be equal to 1 minus the type 2 error we introduced in a couple of slides before. So that's to say, if the type 2 error is smaller, there will be a higher power. And there's a relationship between power and sample size. And we usually want to know, how is the sample size related to statistical power? First of all, remember, in the previous block of this lecture, we talk about that standard error generally decreases with the rate of 1 over square root of n. That is, a large sample size lead to a smaller standard error. And statistical power is inversely correlated with the standard error. That's to say, a smaller standard error or greater precision corresponds to a higher power. If you relate the first and second relationship, and you can conclude a higher power can be attributable to a higher or larger sample size. In summary, hypothesis tests are used to test scientific hypotheses by utilizing sample parameters with a decision rule. The higher the sample size for a given hypothesis test, the smaller the standard error, and the higher the power of the test to detect a true positive test result, what we call the signal. That's all for this block, and this is the end of the statistical inference lecture.